Prokaryotic cells have a high concentration of dissolved solutes in their cytoplasm, making their cytoplasm hypertonic to the environments that the cells live in. The tendency is for water to flow into these cells, and without a rigid support, the cells would burst due to increasing volume. The cell wall provides the rigid support that prokaryotes need for survival. It's also an antigen and a drug target for antimicrobials. This lecture on the cell wall is one of the most important that you'll have during the semester. We're going to lay the groundwork for understanding the structure of the bacterial cell wall and its importance to human health and medicine. By the end of this video, you should be able to describe the function of the bacterial cell wall. You should be able to explain the major characteristics and differences between gram-positive and gram-negative cell walls, and you should be able to explain the medical significance of lipid A. So here's the table. Remember, I told you there would be a table that I'd share with you that compares prokaryotes and eukaryotes. For this particular video, we're going to talk about the cell wall and energy metabolism. So your prokaryotes have cell walls usually, not always, but usually. About 90% of bacteria have one, and almost as many archaea have one. Bacterial cell walls tend to be made out of peptidoglycan, and archaeal cell walls tend to be made of pseudomurine, which is like a cheap knockoff of peptidoglycan. Eukaryotes, on the other hand, don't have cell walls that contain either of these molecules. Instead, eukaryotic cell walls, like those we find in algae, plants, and fungi, they tend to be carbohydrate-based, made of chitin and cellulose. For energy metabolism, we're really talking about where ATP synthesis happens. For prokaryotes, ATP synthesis happens in the cytoplasm of the cell and at the plasma membrane. In eukaryotes, energy synthesis usually happens in the cytoplasm, but also in the mitochondria and chloroplasts. You'll remember that in these videos we've been on an odyssey, traveling through the structure of a prokaryotic cell, starting from the outside and working our way in. We started off with the glycocalyx, which in this figure is labeled as the capsule, but remember you also have slime layers. So the glycocalyx is this kind of brick-colored layer that's on the outside of the cell. As we move into the cell, we encountered the flagellum, which is used for motility, pili, also used for motility, among other things, and fimbriae, the short hair-like structures that are used for attachment by some bacteria. Now, we want to approach this purple layer here. The purple layer is the cell wall. And the cell wall of a prokaryote has three main functions. The first function is it gives the bacterium or the archaeon its shape. So most cells are monomorphic, meaning they're born with one shape, and although they may get bigger, they maintain that same shape. It could be a coccus, a bacillus, a spiral, or something else. It's the cell wall that's responsible for that shape. The second function is that the cell wall anchors flagella to the prokaryotic cell. So remember, at the very base of your flagellum, you have these disc-like protein structures called basal bodies, and they are tightly associated with the cell wall of the bacterium. The third function of the cell wall is it prevents osmotic lysis. As you heard at the very beginning of this video, the cytoplasm of a prokaryotic cell is hypertonic to the environment that the cell is in. So the tendency is for water from the environment to actually flow into the cytoplasm of the cell. Now, without some sort of rigid border surrounding the cell, eventually the cell would just swell and swell and swell and burst, kind of like a water balloon that you put under the faucet and just fill until the balloon can't hold anymore. So the cell wall prevents that bursting due to osmosis. That's why it's called osmotic lysis. The cell wall of your prokaryotes, and we're talking about bacteria here specifically, is made up of an unusual molecule called peptidoglycan. And peptidoglycan is an antigen. That is, it can stimulate the production of antibodies by the human body. This is the structure of peptidoglycan. It's a polymer made up of disaccharides. The sugars that make up the disaccharide are called NAG, or N-acetylglucosamine, and that's represented by the brown color here. So this is NAG. And then the other part of the disaccharide is NAM, which is here, which is N-acetylmuramic acid. And both of these are derived from glucose. They're made in the cytoplasm of the bacterium and then transported out of the bacterium to an area just outside of the cell called the periplasm. As you can probably see from their structures, NAG and NAM look chemically extremely similar. The only difference is here at carbon number 3, we have a bond that goes from the NAM molecules, and attached to that bond is a chain of amino acids. Now, the disaccharide of NAG and NAM alternates to form a backbone for peptidoglycan. The backbones are joined together chemically. We call them cross-linked. We say they're cross-linked together through the amino acids attached to NAM. So you can see here are the amino acids, and this little dotted line, that's the cross-link. It goes from the lysine on one NAM molecule up here down to the alanine on the NAM molecule down here. What this does is it strengthens the cell wall. Kind of like how at a park, you know how there's that chain link fence that usually goes around? And if you've, of course you were a kid, if you've ever played at one of those playgrounds and you've run up against that chain link fence, you know it's really strong. You can put your full body weight on it and it'll support you. So the reason why those fences are so strong is because the links of the chains are interconnected, and that gives strength to all of the links. Well, the same thing happens with NAM in the cell wall for your bacterium. The cross links between the NAMs greatly strengthens the cell wall and greatly protects bacteria from osmotic lysis. Bacteria are divided into two groups based upon the structure of their cell walls and how much peptidoglycan there is. The first group of bacteria are called the gram-positives, and they have peptidoglycan. You can see the diagram of it here. These are the peptidoglycan backbones that I mentioned on the last slide. So you can see we're alternating the NAG and the NAM to make these backbones. The green spheres here represent the amino acids that are attached to the NAM. And the blue spheres represent the crosslinks that connect one NAM molecule to another on an adjacent backbone. And again, this helps to stabilize and strengthen the cell wall. Now in your gram-negatives, we're going to have 3 layers of this peptidoglycan. Here's a diagram showing you 1, 2, 3 layers. And notice that they're all connected via the NAM. So this really helps to strengthen the cell wall. In addition to the NAG, NAM, and amino acids, there's another structure in the gram-positive cell wall, and it is called teichoic acid. There are 2 types of teichoic acid. You have the wall teichoic acid, like this one, which is only found in the peptidoglycan layer. And then you have lipoteichoic acids like this one that are actually connected to the lipids in the plasma membrane. And then these lipoteichoic acids run through the cell wall as well. Now, what the teichoic acid actually does is under dispute, but we think that it's covalently linked to the muramic acids and it stitches the layers of the peptidoglycan together. Thus, it helps to stabilize the cell wall even more and make it stronger. The second group of bacteria are called the gram-negative cells, and they are called gram-negative because of their cell wall. By looking at this diagram, you can see there's some pretty obvious differences between the gram-positive and gram-negative cell wall. So the gram-negative cell wall consists of a lipid layer here called the outer membrane, and then one single layer of peptidoglycan, which is located in this space, which is called the periplasm. So one layer of peptidoglycan is sandwiched between the outer membrane and the plasma membrane of the cell. This peptidoglycan layer is not cross-linked in the same way that it's cross-linked for your gram-positives. Instead of all of the cross-links, the peptidoglycan is actually attached to the outer membrane through these proteins. You can see here and here. These are called bronze lipoproteins, and they have a hydrophobic anchor in the outer membrane, and then they bind the peptidoglycan to the outer membrane. Now, two things you need to know about the gram-negative cell wall. It's toxic, and it makes gram-negative cells harder to kill than gram-positive cells. Here's why. Let's start with why it's toxic. No, let's start with why these cells are harder to kill. Well, let's talk about both. So, your gram-negative cells have an unusual molecule in them called the lipopolysaccharide, and it's represented in this figure by these little squares with the stalk or the antenna going up. It turns out that lipopolysaccharides give a negative charge to the outer membrane, and this is going to help repel hydrophobic compounds like certain drugs and disinfectants that would ordinarily kill the cell. Some gram-negative species live in the guts of animals like us, and the lipopolysaccharide repels fat solubilizing molecules like bile. So this repulsion allows bacteria to survive in places that would ordinarily be inhospitable to bacteria. When lipopolysaccharide, these purple molecules, are released from bacterial cells, they're actually toxic to mammals. So let's talk about that. So we were talking about lipopolysaccharide, and we said that it's medically important because it's actually considered to be an endotoxin. Endo, E-N-D-O, is a prefix meaning inside, and toxin means, well, it's a toxin. And the reason why lipopolysaccharide is an endotoxin is because of this molecule right here at its base. This is called lipid A, and it has a couple of fatty acids attached to it, so that's how this lipopolysaccharide, or LPS, embeds in the outer membrane here. So when LPS gets released from bacterial cells, it creates a variety of physiological reactions. It can induce fever, so endotoxins are said to be pyrogenic as a result. Pyro, P-Y-R-O, genic, G-E-N-I-C, means fever-causing or fever-inducing. The lipopolysaccharide also changes white blood cell counts. It can cause materials to leak from blood cells. It can also lower blood pressure and cause vascular collapse and eventually shock. Remember, shock is a life-threatening drop in blood pressure. Your organs can stop functioning properly and it can lead to death. So that's the lipid A part of this molecule. In addition, you have a core of carbohydrates, a polysaccharide, and then attached to that core, you also have this, which is also a polysaccharide, and it's called the O antigen. Do you remember what an antigen is? Right. It stimulates the human body to produce antibodies, right? So the last time we talked about antigens, I mentioned a specific E. coli strain called E. coli O157H7. And we said that the H7 referred to flagellin, which is part of that bacterial cell. Well, the O in O157H7 refers to the lipopolysaccharide, specifically the O antigen that's at the very end. So we can distinguish different strains of E. coli based upon their O antigens. Remember, a strain is simply a subgroup of a species that has a slight genetic difference that's distinguished by antibodies. All right, so that O antigen is part of the part of the lipopolysaccharide, as is the lipid A. And I don't remember if I said it, so just in case I didn't, I'll say it now. Lipopolysaccharide is called an endotoxin, endotoxin. Endotoxins are toxins that are actually part of the cellular structure. So that's what LPS is. Just a couple more things about the outer membrane. So not only does it have the lipopolysaccharide in it, but it also protects gram-negative cells against phagocytes, complement, and antibiotics. So phagocytes would be cells like neutrophils and macrophages that come along, they bind bacterial cells, and then they engulf them, they gobble them up and digest them. So phagocytes have a hard time binding the outer membrane of gram-negative cells, which means that they're less effective at gobbling up and digesting these cells. Complement is a set of proteins produced by your liver, and they assemble on the surface of bacteria and punch holes in the bacteria. You can think of it as being like a defense system in the human body. Well, some of those complement proteins don't attach very well to the outer membrane, which means that complement can be less effective at killing gram-negative cells. Then there are the antibiotics. So remember how I said that the outer membrane is a phospholipid bilayer, and how you probably remember from your Gen Bio that most large molecules and charged molecules don't diffuse through phospholipid bilayers very easily. Instead, they usually have to have some sort of channel protein that they travel through the membrane. Okay, remember that? You probably talked about it with respect to ions crossing the cell membrane. Similar thing happens with bacteria. Instead of having channel proteins, your outer membrane has what are called porin proteins. So when nutrients need to come into the cell, they have to go through a porin protein to actually get to the cell. The bad thing about this is that if a molecule is too large to fit through a porin protein, it can't actually get into the bacterial cell. And for certain antibiotics, that's the case. They're too large, they can't go through the porin protein, and thus they cannot reach their cellular targets, and gram-negative cells tend to be resistant to those antibiotics as a result. Whew, that was a lot. So this is another representation of gram-positive and gram-negative. Here's gram-positive on this side. How do we know? There's multiple layers of peptidoglycan. We call the gram-positive cell wall thick because of the multiple layers of peptidoglycan. Notice that this figure also shows you teichoic acids. Here's the gram-negative cell wall. Again, just another representation. This is the plasma membrane for the gram-negative cell here. This is the outer membrane. So there's one layer of peptidoglycan here. It's attached to the outer membrane via special brawn lipoproteins. Embedded in the outer membrane, we have porin proteins like this one that allow molecules to pass from the outside of the bacterial cell across the cell wall and into the cell. Okay, and you have that lipopolysaccharide that I mentioned. That is an endotoxin. So when gram-negative bacterial cells are reproducing inside of the body, some of that lipopolysaccharide and lipid A actually gets released, and it causes people to run a fever. It's one of the reasons that you know you're having an infection. Whether a cell is considered to be gram-negative or gram-positive is because of its cell wall. And we determine what kind of cell wall a cell has by doing a chemical procedure called the Gram stain, which I'm going to talk about in a different video. Gram-positive cells will stain purple with a Gram stain, and gram-negative cells will stain red or pink with a Gram stain. And the reason why these Gram designations are so important is because if we do a Gram stain, let's say a patient comes in, they've got a number of symptoms. A doctor anticipates that this person has a bacterial infection. If a specimen is taken, let's say from an abscess that the person has, if the specimen is taken, you can do a Gram stain in about 5 minutes, give or take, and know something about the structure of that bacterium. And that structure is related to what types of antibiotic treatments you can do, resistance or susceptibility to disinfectants and antiseptics. It's related to resistance to physical force. And this kind of gives clinicians a heads up on what types of treatments might be effective against those particular bacteria. So this table summarizes some of the information about the structure of the cell wall as well as resistance or susceptibility to different factors. So let's go over it really quickly. Again, your gram-positive cells, they're called thick. They're said to have thick cell walls because there are multiple layers of peptidoglycan. There is no outer membrane, and since there's no outer membrane, that means there's no lipid A or lipopolysaccharide. Because these cells have a thick cell wall, they're resistant to physical force. So if you think about, say, a dialysis machine or some other machine in a doctor's office, many times they'll have these cleaning cycles where disinfectant or high pressure water is pushed through a series of tubes. Well, if you're dealing with a machine that has been contaminated by gram-positive bacteria, the bacteria may be less likely to be killed by that disinfecting wash, just by the pressure of the chemicals running through the machine. Lysozyme is an enzyme that's in your tears and in your sweat. It breaks the bond between the NAG and the NAM and peptidoglycan, so it can weaken the cell wall. Lysozyme on our skin is protective against infection by gram-positive cells for this reason. These cells are resistant to drying out because of the thick layers of peptidoglycan. They kind of insulate and hold water in the cell. And these cells are resistant to antibiotics like streptomycin, tetracycline, and chloramphenicol. Let's look at your gram-negatives. Your gram-negative cells, as you know already, have one layer of peptidoglycan, and they do have that outer membrane. Remember, the outer membrane and the lipopolysaccharide that's embedded within it makes gram-negative cells resistant to a lot of disinfectants. It turns out that because of the lipid bilayer of the outer membrane, these cells are also resistant to certain antibiotics like penicillin because the antibiotics are too big to fit through the porin proteins, and they also don't diffuse through the lipid bilayer, so they're excluded from the cell. Lysozyme is a large protein, also doesn't fit through the porin proteins, so that means that lysozyme is also not effective against your gram-negatives because it can't actually get to the peptidoglycan to have any effect. Your gram-negative cells have that lipid A, so remember lipid A is an endotoxin. It's responsible for different physiological effects in the body. These cells, because they only have one layer of peptidoglycan, are sensitive to physical force. They are sensitive to drying out, again, because they don't have a lot of insulation to hold water in the cell. And they are sensitive to antibiotics like streptomycin, tetracycline, and chloramphenicol. So we get all of that information just by knowing how certain cells stain with the gram stain. So that's it for this particular video. What I hope you got out of it was that the cell wall has three main functions. Number one, it anchors flagella. Number two, gives bacteria their cell shape. And number three, prevents osmotic lysis. Remember, osmotic lysis is when cells burst open because of osmosis. So the cell wall provides a rigid barrier that prevents a cell from expanding too much as water flows into it. I also gave you the main characteristics and differences between your gram-positive and your gram-negative cells. Hopefully, somewhere in your mind, you will remember the structural differences between these two types of cells. And you'll also hopefully remember that your gram-negative cells are typically harder to kill because they're resistant to antibiotics, they're resistant to certain disinfectants and antiseptics. Okay, and I hope you'll remember that gram-negative cell walls are called toxic because they have that endotoxin called lipid A. We also talked about lipopolysaccharides. They are antigens that the human body develops an immune response to. So that's it for this video. In the next video, we're going to actually talk about the gram stain, and we'll find out why the gram-positive cells stain purple and the gram-negatives stain pink. The last thing I hope that is clear to you is that if we know how a cell stains with the gram stain, there's a lot of other information that we can infer about that cell, and we might even be able to determine what types of treatments could be effective against the cell. So that's it. I'll see you at the next video. If you have any questions, please reach out to me by email or text. I'm not really in my office these days, so please don't use the phone number, but email and text are great. Talk with you later. See you at the next video.